In this tutorial, we'll walk through the new revised main document for the Genome GPS DNA sequencing workflow. This essentially serves as a one-stop shop for all your quality control metrics needs and your initial starting point for tertiary data analysis. Briefly, as we walk through the Genome GPS, the first thing that we see is a little bit of sample information. This is important to track what samples were actually included in a particular run, what type of disease it has, and how many samples. We also maintain information that's very detailed that we can always go back and recreate the data that we've generated by this tool info file, which is basically a list of every tool that we used and any parameter settings that are included. We also track which capture kit was used, if it was a capture, and the date that it was run. As we scroll down, we can see the folder structure of where this main document lies, which is represented here, and there are also subfolders in this directory. Now, we've taken the advantage of this HTML format so that you don't have to actually dig in through each of these folders and files. You should be able to access most everything you need via this interface. But you can see we have a lot of information delivered as part of our standard workflow. So let's quickly move down to the results. And in this section, you can see we have a series of panels, and each one of these is clickable and they do different things. For instance, this is the sequence quality metrics. So this is how good the sequencing actually worked. So if we were to open that up, we can see for each individual sample, we can look at the read distribution. So each one of these is the average read quality score per base. This is a 100 base pair run, and you can see they're very high. You can also dig in deeper by clicking on that. We can actually get the reports from a tool called FastQC. But honestly, if your data has got to this point, it's already past Bruce's validation as well as our validation, and you probably won't have to worry about that unless some other questions come up later. So we'll go back to the main page, and next I want to look at alignment metrics. So this is a typical coverage plot that we see. These are two samples that are in this particular data set, and we're looking at their coverage profile. Now, you can't really see at this zoomed out view what the X and Y axes are, but we can when we go ahead and click into this. So as we drill down, we can find out this is the total read alignment metrics. For instance, this particular sample had 98% of reads mapped with a 20% duplication rate. And as we look at the image plot, you can see this coverage profile. The way to read this is on the y-axis is the percent of the kit or the capture region that you're looking at. So it maxes out at 100%. And the x-axis is what level of coverage. So for instance, there is 30% of the target region covered at least 300x. Or at 100% of my targeted region, I have at least 10x. So we go back, and we have mutation results. Now these mutation results are separated by whether or not this was germline data or somatic data. So if you have tumor normal pairing, we have two combinations, or else you just have one. And what these are, as I open up the germline results, is it's from a tool called SNP-Effect. And what it does is it does various QC metrics. It tells you how many variants you're observing on each chromosome, so you can see if you don't have any variants on a particular chromosome, then something has failed. And it also breaks down the different types of effect and their frequency. However, these numbers can be a little bit misleading because we have not filtered the data in any way. And in a lot of cases, in custom captures and exome captures, we tend to see a lot of downstream and intronic variants. Usually you'll want to filter those out because they are of lower quality. and they're also not usually what you're interested in. But it has a lot of information that you can peruse at your leisure. So the same thing for the somatic variants. We do run different tools for the germline analysis and the somatic analysis, so you will have two different reports based on those. So we go back to the main page, and we go down. In Genome GPS 2.0, we've released a number of structural variant modules that will be run. And in this particular case, we did have a copy number tool run. Now, the number and type of structural variants tools that get run is dependent on your experimental design, and we'll have collected all that information at the beginning. So in this one, the only one that was done here is for this particular sample. It was a somatic analysis, so we're looking at copy number changes. And very quickly you can see that there is copy number loss in this chromosome, focal amplifications here and here, and possibly a deletion. There is more fine-scaled results in the actual folders, but this gives you a very quick overview of what your sample actually looks like. So now let's open up the IGV sessions. What we've tried to do with this page is to make it simple for the user to actually look at the variants in their particular samples. And we've divided it up one of two ways. You can either click this link, which will bring up all of the samples in a particular flow cell, which you may want to do in this case because we only have 2 samples. But if you have 100 samples of custom capture, that may not be what you want to do. We also break them up if you do a somatic type analysis. We break them up by individuals. So in our example, we have 2 individuals, but we run a somatic analysis, so we end up getting 1 IGV session per group. And so I'm just going to go ahead and click on this group ID, and what that's going to do is it's going to cause me to download this file, and I have to tell my computer that I want to keep this file and then I want to run it. Because I use Chrome, so it has these extra features built in. And what it's doing now is it's actually downloading IGV, and I'm going to tell it that it's okay that it runs. And what IGV does is it allows you to actually visualize your results. Now, because we have these stored on the archive, we actually have to log in again. And when we log in, we actually have to the prefix mfad / and then your M number. Okay, and then I'll just type in my password. Now this will take several minutes to upload, but once it's uploaded, it's actually relatively fast to go from position to position. And so what I recommend is once you've identified variants from later in your studies, you want to go back and validate them, looking at IGV and making sure that the reads are of considerably high quality. So after a minute or two has passed by, IGV now comes up, and it actually has the samples that we have in our data set over here on the left. And we're zoomed out looking at the whole genome, so it's not going to show us anything. But let's just go ahead and zoom in on any particular gene. Let's pick NR0B2. Whoop. NR0B2. And just go to that gene. It's a very small gene, so you can very quickly get a sense of what the data actually looks like. And I apologize, it's running slow because it's accessing Arizona and I'm going through a couple of different levels here. It should be a little bit faster for you. So now we have the coverage data for NR0B2, and each of these are the individual reads, and there's all sorts of metadata associated with those. Now, teaching a course on IGV is a whole something that takes a long time and at least another hour, and so we won't go through in detail. But you can navigate around, and it's a very, very useful tool. So that's all for the imaging. So let's go back to the home page, which you can also get back to by clicking that Genome GPS. And we've gone through all of these with the exception of viewing the results in BioR Web. So BioR Web is our tool that annotates variants. So usually we call single nucleotide polymorphisms or somatic nucleotide variations, and also small insertions and deletions. And although the regular workflow outputs a VCF, which is our final end product, we also know that a lot of people want certain types of information. And so we've had a collaboration with the IT group, and we built this tool called BioR Web. So let's go to BioR Web and see what that is. Okay, after coming to BioR Web, the first thing I have to do is log in, giving it my username and password. And because I've linked directly to BioR Web from that particular page, I've already uploaded essentially the VCF file that I'm wanting to look at and annotate. And over here on the right are several different annotation sources or types of information that we want to put on our somatic variants in this case. And so the first thing I have to do is give it a job name. And so this is I'm going to call this test.somatic. You can name it anything that you want. This helps you remember what exactly the file is rather than some sort of cryptic name that has the flow cell ID and the lane ID and etc. So I'm going to call this test.somatic, and there are these different columns that have different types of information, and they're all selectable by these checkboxes. You can either select them all or just one or two. It doesn't really matter. It's whatever your preferences are. And they're divided up based on the kind of information that they provide. So for instance, this minor allele frequencies is variant specific, so it pays attention to the reference and alternate alleles. And I have a number of different annotations here. Here, for instance, is 1,000 genomes allele frequency in Asians, or Americans, or Africans, or Europeans. I usually like to have allele frequencies from different sources. There's 1,000 genomes, BGI 200, exome variant server, or exome sequencing project, which is 6,500 exomes, and of course HapMap. So all of these tend to be very important for me when I'm trying to find those variants of some statistical significance. If they're common polymorphisms, I'm usually not interested in them depending on the design. There's also information about the variant, such as what sort of gene is it in. Does it have an official gene symbol or an entree ID? Sometimes you want the full gene name because it's more descriptive sometimes than the gene symbol, and whether or not it's in OMIM or causes disease, or if that variant is actually in a microRNA binding site. We could also look at functional predictions. So these are the translational effects of a particular variant. And you can see that they're usually prefixed by either VEP or SNP effect. These are two of the very commonly used annotation tools. And these tools actually work in the background. So you actually don't know how, you don't have to know how to run VEP or know how to run SNP effect, and you can get these types of variants. So for instance, if I want VEP to call the cDNA position, the coding sequence position, the protein, the amino acid, and I want SIFT terms and polyphen terms and scores, I want to know what sort of protein domain the variant is in. All of this I can select, and I already have SNP effect, so I'm not going to select those here. And then we have a couple of tracks over here from UCSC, like is it a transcriptional start site, transcription factor binding site, and there will be another tutorial coming up on BioR Web and a little bit more detail about each one of these different annotations. The other thing that I like about BioR Web is you're able to save these defaults. So you can save this particular profile, which saves the checkboxes, and you can actually give that a name and come back to it from time and time again. So This is actually the one that I prefer to use all the time consistently. So you can see which ones I have selected here, but you can create your own. Or you can load the default, but I'll choose to keep my own. And because I already have the name and the FTP location was already added for me, all I have to do is click Get Annotation. And what happens is I get this pop-up box that says I have submitted this particular file, and an email will be sent to me when the annotation is done. Now when that gets done, I'll actually have a link in my email that I could download directly. Plus it'll actually pop up over here on the left hand side. So what this is a list of all of the VCF files that I've annotated with BioRWeb in the past, and it gives me a link where I can download them. So let's just get a peek for what the downloads actually look like. So again, I have to log in because everything's password protected, and I get a result file which I'll go ahead and open. And we've tried to make it so it'll automatically open in Excel. Sometimes it will give you this sort of warning that says it's not really an Excel file. You just say, yeah, that's fine, I don't mind. And the first thing that you'll see is all of these comments, and these comments are marked by this double hash. Now that's very essential for bioinformatics because that tells us a lot about the file itself, but it doesn't really help the investigators figure out what exactly they want. So we can actually get rid of those because now we're manipulating the file on our own desk drive, so it's not actually affecting the final results. And now I just have the different headers. So these are my two samples. And so basically from this point on, I have a VCF file format. And from this point on, I have all the annotations that I requested for in BioRWeb. So if I only had 2 annotations that I wanted, it would stop here. But of course, I added in a lot. And so as we scroll around, you can see that there's a lot of information that's present here. This actually helps us where we don't give everybody the same 100 columns of information. You can pick and choose which ones you want, and of course that makes understanding what you want a little bit easier, and you're not drowning in annotation that you don't need. So that's BioRWeb. So going back to the home page, the main document, we've now gone through the details of all the things that you absolutely have to know, but sometimes that doesn't always do everything that you want it to. So we do have links to our Genome GPS website, which is right here, and it actually takes you to the current release, and you can get more information about what actually goes in here. We have a technical sheet if you want to get into the really nitty-gritty details, and all sorts of other information about previous versions. We also have 2 other features that may be of interest. If you want further secondary and tertiary type analysis, you can get help through our IRC pipeline. And you just click that and it takes you right to our IRC page, and you just fill out a little bit of information, and we can get back to you about what exactly you're looking to do and how we want to do it. We also have a mailing list that you can join for Mayo, where any sort of announcements where we've upgraded a particular workflow or we've got some sort of insight that may be useful to you. So you can see in this short amount of time I've already got my results back from BioRWeb. It says that that particular sample ran successfully. This was how many variants went in, and this is how many variants came out. And of course here is my link that I can download. If you ever have any problems with BioRWeb or you don't have any understanding, we also have this link in the email that it sends after you've submitted a job to BioRWeb that will take you to a remedy ticket. And I have to log in for this as well. And this automatically fills out some things for you. And you just put in what sort of problem that you're having in this field, and click submit, and we'll get back to you as soon as we can. So hopefully this was informative to you, and I hope that it streamlines your analysis process. And if you have any questions, just let us know. Thank you.